Bismillahirrahmanirrahim. In previous lecture, I discussed about introduction of steroids, types of steroids, and the properties or application or uses of steroids. In this lecture, I will discuss how we synthesize steroids, cholesterol, and other steroids. Actually, I am going to discuss their biosynthetic pathway. So first, I will discuss how cholesterol is synthesized in body, and then how other steroids are prepared. So topic is biosynthesis of steroids. As we know that glucose undergo glycolysis, pyruvic acid is formed that upon oxidation give acetyl coenzyme A, and this is also obtained from fatty acid oxidation and other sources. So acetyl coenzyme A is the first basic unit which start cholesterol synthesis. 2 acetyl coenzyme A, these are condensed in the presence of thiolase enzyme. 1 coenzyme A is removed and the 4 carbon structure acetoacetyl coenzyme A is formed. Another acetyl coenzyme A condense with acetoacetyl in the presence of hydroxymethyl glutaryl synthase enzyme, and we get 6 carbon structure. If we number the carbon 1, 2, 3, 3 hydroxy, 3 methyl glutaryl coenzyme A. This compound undergo reduction in the presence of HMG reductase enzyme. Coenzyme A is removed and NADPH is a source of hydrogen and electron, and we get mevalonate 6 carbon acid. Mevalonate, it is phosphorylated, 2 phosphate added here and 1 here. 3 ATP molecules are changed into 3 ADP and we get 3 phospho, mevalonate, 5 pyrophosphate. Phosphorylation occur at 3rd position and 5th position. This pyrophosphate, it undergo decarboxylation, and this phosphate is removed in the form of ATP, and this carbon dioxide is removed and we get isopentenyl pyrophosphate. It undergo isomerization and give dimethylallyl pyrophosphate. So this dimethylallyl react with isopentenyl pyrophosphate formed here. This is 5 carbon complex, this is 5 carbon compound. So these two are added with each other. 2 inorganic phosphates are removed in the presence of geranyl pyrophosphate synthase enzyme, and we get 10 carbon compound geranyl pyrophosphate. This compound react with another dimethylallyl pyrophosphate, and in the presence of farnesyl pyrophosphate synthase, and after this addition we get 15 carbon compound farnesyl pyrophosphate. It undergo a series of reaction and ultimately we get sequelin which contain 30 carbon, and the enzyme which catalyze final reaction is sequelin synthase. Sequelin undergo oxidation in the presence of sequelin 2,3 oxygenase to form sequelin 2,3 epoxide, which after a series of reaction is converted into lanosterol, which again after series of reaction changed into lithosterol, 7-dehydrocholesterol, and finally we get cholesterol 27 carbon compound. So rest of the steroidal hormones or steroidal structures are prepared from the cholesterol. So this is the biosynthetic pathway of cholesterol. Now let us discuss that how other steroidal hormones are prepared. So first of all, let's see how progesterone is formed. The cholesterol, this enters into the mitochondria, and in the presence of P450SSC complex, it is converted into pregnenolone. So what changes occur? This carbon undergo oxidation and this chain is removed and we get this structure. This isomerize into the progesterone. So first steroidal hormone, female sex hormone progesterone is prepared through this pathway. Okay, now let's see how aldosterone is prepared. The progesterone formed undergo hydroxylation over this carbon. Here hydroxylation occur, rest of the structure is same. Rest of the structure will remain same and progesterone is converted into deoxycorticosterone. This deoxycorticosterone undergo hydroxylation in third ring of the steroidal nucleus. Rest of the structure will remain same as previous and we get corticosterone. This corticosterone undergo oxidation at this carbon represented in red. So the changes which occur during each reaction is highlighted with the red pointer. So ultimately we obtain aldosterone. So two steroidal hormones are prepared, one progesterone, other is aldosterone. Okay, now let's see how estrogens are prepared. So progesterone formed from the isomerization of pregnenolone is undergo hydroxylation at 17 carbon. Rest of the structure remains same and we get 17 hydroxy progesterone, which undergo oxidation at 17 carbon. So we get androstenedione. This structure, rest of the structure attached with this 5 carbon ring is same as this. And this androstenedione undergo hydroxylation and aromatization of the first ring of the steroidal nucleus as highlighted by this red pointer and we get estrone. This estrone undergo reduction at this carbonyl carbon and ultimately we get estrogen or estradiol. So how estrogen is prepared from cholesterol? First cholesterol is changed into pregnenolone, isomerized to progesterone, and then this progesterone undergo changes to 17 hydroxy progesterone and then androstenedione, estrone and then estrogen. So how cortisol is prepared? 17 hydroxy progesterone in the presence of P450C21 undergo hydroxylation at this carbon. Rest of the structure is same and we get deoxycortisol. This deoxycortisol undergo hydroxylation at third ring and we obtain cortisol. So how cortisol is prepared from cholesterol? We can follow this pathway. Finally, how testosterone is prepared? Pregnenolone undergo hydroxylation at this carbon and we get 17 hydroxy pregnenolone. This in the presence of lyase, oxidation takes place at carbon number 17. Rest of the structure is same as this and we get dihydroepiandrosterone. This undergo isomerization and we get androstenediol, and this in the presence of testosterone synthase undergo changes at carbon number ring 1 here and we get testosterone. So this is all about the biosynthesis of steroids in the body. Thank you very much.